here. Starting out with a real brief kind of intro to chapter 13. First, what I want you all to know is, according to at least the study guide, the format will be the same for the test. There'll be 14 multiple choice, 30 fill in the blank, and 4 written answer questions. And all the constants, equations, and concentrations will be provided. So you won't have to memorize, the specific equations, especially with chapter 13, and the constants as well. But there will be some calculations. So I would recommend bringing a calculator, whether that be a scientific or a graphing calculator. Does anyone have any questions about those, about kind of the brief layout of the test? Okay, cool. So I'm just going to do like a quick run through of what we're supposed to know, or what you guys are supposed to know for chapter 13. Chapter 13 will only have 3 questions, at least pertaining to it. So I won't spend like a whole time on this. Most of the time will be spent on chapter 14, chapter 15. Jason said he won't be able to make it today. His birthday's today, so he wanted to spend time with his wife and everything. So please bear with me. I don't know if Rebecca will be joining in, but if I'm the only one, then just bear with me throughout the next couple of hours, I guess, the next 2 hours. I'll try to do my best to answer questions you do have, and cover the material that will be included on the test. So with chapter 13, the first bullet point says, know how to calculate delta G prime naught when combining reactions, or how to calculate KQ from the delta G, or calculate delta E from the 2 half reactions, and how to convert delta E to delta G, and understand when these terms are used. Can anyone help me or help explain what delta G is? What was delta G or delta G prime naught? Does anyone know what that is?
It's just the change in free energy under standard conditions over the course of a reaction.
Mm-hmm. Yeah. So it's the Gibbs free energy, and it is the kind of like the standard free energy of things, or standard free energy. So this delta G prime naught is classified as the free energy change under standard conditions. So under these conditions, it's 1 molarity, 25 degrees Celsius, and at a pH of 7. So this number, or this delta G, does not change. There's usually just like one under these conditions. There's only 1 delta G for one reaction, because a reaction under these conditions, there's only one certain delta G prime naught. A delta G in this case, just like a regular delta G, is the Gibbs free energy of an equation, or a reaction, not under standard conditions. So there's no equilibrium in the cell, and so this is the formula that you'll be kind of going off of if you want to find what delta G is. You can still find delta G prime, but they would just have to give you this equation, or the values, at least some values for this. They'll give you like the concentration, or in summary, they'll either have to like give you like the KQ, or just like some of these values in order to calculate what this number is. Does anyone have any questions about Gibbs free energy, things like that? Okay. We'll just go ahead and just keep going along here. That equation that we were just looking at, Delta G when it's negative is spontaneous. 0 is when it's at equilibrium, and above 1. Sorry, this is KEQ, sorry. This is just the relationship with that, sorry. When delta G is negative, it's spontaneous. When it's positive, it's non-spontaneous. This is just coupling reactions. This is important to know for the exam of knowing how to couple the reaction. So what we have here is we have a equilibrium constant. So overall, the equation for this reaction is glucose plus phosphate is equal to this, and the delta G prime naught is plus 13.8 kilojoules. So if we want to find out what at least kind of what KEQ is they're trying to solve for KEQ. So they're just substituting this. So at KEQ, at KEQ, delta G is 0 because it's in equilibrium. And so delta G prime naught is equal to this equation. Let's see. This is what occurs when it's being coupled. Delta G. That's pretty much what 13.1 is. I'm just going to transfer quickly to 13.2. So the other bullet points include learning how to recognize redox reactions and how they take place. So on 13.2, it's a good slideshow to review to understand how to recognize redox reactions. And so in this case, a species that loses electrons is being oxidized, rather a species that gains electrons is reduced. Kind of like with glycolysis in step 6. Step 6 is an oxidation reaction because your overall the species, the glyceraldehyde 3-phosphate is being oxidized, which means it's giving away electrons to NAD+, and NAD+ forms NADH in that reaction. So even though NAD+ is getting reduced, glyceraldehyde 3-phosphate is getting oxidized. And mostly it's just trying to, at least like involved with 13.1, is just usually recognize what's getting reduced and what's getting oxidized. Usually a key for me, like when I'm recognizing reactions that are being reduced or oxidized, I usually just look to see if it involves NAD+, FAD, or CoA. Because usually one of these will either get oxidized or reduced. And when you see that, then you kind of know instantly, okay, this is a redox reaction. That's usually kind of like a hint for me whenever I see that. And then as far as the last one, know about general strategies for enzyme regulation as part of glucose and glycogen metabolism. That one I believe can also be found, I think that was in 13.1. Let's see if I'm double check this real quick. Yeah, regulation of metabolism. So it says know about general strategies for enzyme regulation. So these are just different ways that enzymes can be regulated through transcription, synthesis, release, catalytic activities, phosphorylations, allosteric regulators. These all regulate enzymes. So this would be a good slide to know for that one. So any questions about 13.1? Or sorry, not 13.1, but overall with chapter 13. Kind of just wanted to go by real quick with it, just so you guys can have a quick review. Okay. Let's go on to chapter 14. Now, this is the mother lode of all the questions that that Dr. Graves will be providing. Let's see here. You pull up 14.1. Okay. So glycolysis. So with overall with chapter 14, Dr. Graves expects a lot of you guys to know about glycolysis, especially the details involving the step-by-step reactions, as well as the enzymes and kind of how they're regulated as well, like special features. So we said like know the substrate and product for the enzyme, including the names, structures, know the enzymes, their names, and as well as their reaction mechanism. So so there's a question on the test where it asks you to draw out the reaction or the electron mechanism of how, like, you know, kind of like how OCHEM was when you kind of drew how the electrons would transfer to different atoms. And so that's kind of what he's expecting for you all to know. I know that he covered some of the reaction mechanisms in class. Special features. So the special features kind of include the active site, the critical site groups, and the cofactors. And so I'll kind of like make mention of several special features of the enzyme. I don't think we'll have enough time to go through every single enzyme in glycolysis, because I kind of want to review a little bit of chapter 15 as well. But I'll kind of point out to you all kind of like what he's referring to as far as so. Yeah, and then he recommends just to consult the sets of slides, animations, and content. Animation can also, I know he mentioned, or he put, I was checking Learning Suite today, and he put several animations of each of the 10 steps of glycolysis under the content section in Learning Suite. So you do have that as one of your resources to kind of review. So, so glycolysis, no, it's overall just a reaction where you go from glucose to pyruvate, and I'm sure you guys all know that the overall, it produces 4 ATP, but there is 2 ATP that's invested in it, so you have a net gain of 2 ATP. This is kind of just like an overview of the glycolysis. I think I'm going to go a little bit towards about talking about like the enzymes. Oh yeah, this key right here, it's good to know, it's important to know that 3 out of the 10 reactions, of course, have the large negative delta G's, which means that under these conditions, it's irreversible. So there's 3 of those enzymes, or 3 of those steps, and that's steps 1, 3, and 10. And each of these are regulated by allosteric effectors such as like ATP. ATP is kind of acting like almost as a cofactor because it's causing the enzyme to be activated. There's also like covalent modification, phosphorylation, at steps like 1 and 3. There's also like also increased copies of enzymes. I think one of the questions on quiz 4 talked about that, as well as cell location. Is important to know which steps, where glycolysis happens. And does anyone know where glycolysis happens in the cell?
It's in the cytosol, right?
Mm-hmm. Yeah, exactly. So this is just step 1. Kind of what he was referring to as far as the kind of like special features of the enzyme. So on hexokinase, hexokinase's purpose is to convert glucose to to glucose 6-phosphate. And it does this because it has, uses this ATP and this magnesium ion. This magnesium ion stabilizes the phosphoryl groups that's on ATP. So it allows those to be stabilized so that one of its phosphoryl groups can then detach from ATP and then bind to the carbon or the 6 carbon on glucose. And so also mannose and fructose, we'll talk about this a little bit later, but in other feedback, I believe in chapter 15, maybe it's in chapter 14, real quick. The feeder pathways, that's what's referred to. So in different feeder pathways, hexokinase can also donate, or can also cause like a phosphoryl group to be donated to either mannose or fructose. And we'll kind of talk about that a little bit later. But it's important to know that with hexokinase, a large induced fit occurs with substrate binding. And these bullet points kind of talk about what's going on with this large induced fit. And so these pointers are important to know. With this case in the hexokinase, the 2 lobes closed envelops the ATP. So an induced fit means that the molecule kind of weaves its way through the enzyme. So the fit is kind of induced. So you can imagine this large enzyme, this protein, and each of the cofactors kind of weaves its way through, and it has an induced fit within the enzyme. So that's kind of what it's referring to with induced fit. And the majority of enzyme substrate reactions occur that way, kind of like how oxygen moves through hemoglobin. So the 2 lobes closed envelops ATP and glucose, and there is an 8' amps movement. So that's important to know as well. So the active site environment becomes nonpolar. Usually sometimes the active site is a little bit polar because it's more on the inside of an enzyme. And of course the cytosol is very aqueous, but in this case, the active site environment becomes nonpolar. And it facilitates a nucleophilic attack. Proton, this kind of talks about what's going on with the reaction mechanism. Proton on that, the 6 carbon removed prior to attack on the terminal phosphorus. So this oxygen group attacks the phosphoryl group of ATP and excludes water and prevents simple ATP hydrolysis. So kind of prevents ATP from breaking up. Enzyme catalysts after both that induced fit common features of kinases. A key here with a lot of these enzymes, if you forget kind of what the role of an enzyme is, sometimes you can tell what the role is just by looking at whether it's a kinase or it's a phosphatase, a hydrolase, an isomerase. All of those are keys to you to tell kind of what function the enzyme performs. So a kinase always puts a phosphoryl or phosphate group on something. So in this case, it's putting a phosphate group on glucose. So that glucose becomes glucose 6-phosphate. In step 3, also is a kinase that puts a phosphoryl group on the fructose molecule. I think it's the 6-phosphate fructose. In step 7, it produces an ATP. It takes away a phosphoryl group from 1,3-biphosphoglycerate and puts it on an ADP molecule to form ATP. So kinase is always adding a phosphoryl group onto something. A phosphatase removes a phosphate group. Sometimes it can be kind of confusing. You can say, oh, well, phospho is included in the enzyme, but phosphatase removes some phosphate groups off of molecules. A hydrolase, involves the process of hydrolysis, which is the breaking up of water. A dehydrogenase is an enzyme which allows removal of a hydrogen atom. An isomerase, converts an isomer of a molecule into its other isomer form. or I guess moves isomers. Transferase moves a functional group. So you have all of these different clues to kind of tell you what these enzymes do. Does anyone have any questions about those? I kind of went on a little bit of a rant there. Okay. Here we have step 2 going on. Here it's a key isomerase. So what we know from this is that it's kind of just moving, it's neither adding electrons, not detracting electrons. It's not really adding groups or transferring groups. It's just basically kind of like reforming the group here. So glucose 6-phosphate kind of reforms to fructose 6-phosphate. Some special features of phosphohexose isomerase, occurs in a dimer. Must accommodate negative charged phosphates. So this is a reverse reaction. As you can kind of tell, the delta G prime naught, it's positive, so it's, can convert back and forth. The rearrangement, know the reaction mechanism with this. I believe slides 17, 18 kind of go through how the reaction mechanism go. I'm not going to go too much into detail about that because a lot of these slides kind of tell you how the reaction mechanism goes. But it's just important to know how the reaction mechanisms of these enzymes go through. And a lot of these reaction mechanisms are pretty similar. So like steps 1 and 3 can be kind of similar because both of them are just adding a phosphate group onto. So a lot of those reactions can be similar. I know that step 10 is a reversal of step 1. So you have different clues here. One of the strategies I kind of used when I took the class was kind of noticing the patterns of reaction mechanisms, noticing, okay, what's similar with these steps. So it's just important to kind of know how that goes. So we have step 3, phosphofructokinase. How much details do we need to know in terms of mechanism? I would recommend knowing the arrow pushing. I know that there's a question that's on the test that asks you to describe the reaction mechanism of one of the enzymes in glycolysis. So I would recommend knowing kind of like the arrow pushing of the electrons. And so that'd be good. You don't need to know the delta G for the steps. That'd be kind of brutal. So, but knowing, of course, knowing which steps are reversible and which ones are not, because that kind of plays a role in how the enzyme works. Usually what I did to kind of practice the reaction mechanisms is that I would I would get like a whiteboard, and then I would just get like just grab like a whiteboard, just go to like a class in the library or something, and just draw out repetitively the different reaction mechanisms so that I could secure it in my head. And that way, I didn't waste any paper or things like that, but that's just kind of like what I did to kind of, just to get it down. Phosphofructokinase, very important enzyme. It's another one of those irreversible enzymes. Has a large negative delta G. And, we're just adding a phosphate group here. The reason I want to touch base on this one is because this is it's exergonic. It's also the first committed step of glycolysis. So when a reaction, does this, when the glycolysis goes through this step, it's like a all or nothing type of response. Excuse me. So as a result, this is often the rate limiting step of glycolysis. And these are all just different factors that, these are factors that inhibit or downregulate this enzyme. And these are all different cofactors or different parts that activate it or upregulate it. And slideshow 15.1, it kind of talks about the upregulation and downregulation of glycolysis enzymes. So we're going to touch base on that mostly in like the second hour of the review.
Josh,
Yeah.
I just want to clarify, does highly exergonic mean that it has a really low delta G?
Yes, yeah, very negative delta G. Exergonic kind of means like a large release in energy or a lot of release in
Okay.
heat.
Cool, thanks.
Yeah, so that just kind of helps contribute it to being irreversible. Yeah, so this exists as a dimer of dimers. What's a dimer of dimers? Do we know what a dimer of dimers is?
So isn't it kind of like a tetramer, but the tetramer can be separated into like two halves as well?
Mm-hmm.
It's like you have two dimers that form, and then those two dimers bind.
Yeah, exactly. So the slideshow 24 kind of talks about, or kind of, I guess, like depicts what it's referring to as a dimer of dimers. Hemoglobin is also a dimer of dimers because, it has two alpha beta subunits, or at least, yeah. Total, see, two alphas, two betas, and it's just, yeah, like two dimers just binding to each other. It has two ATP binding sites. First is that's the substrate, and this one acts as the allosteric regulatory site. And this kind of describes what's going on with that. So aldolase, aldolase is step 4, and aldolase is a kind of another key enzyme. This one has a very unique reaction mechanism because it's literally cutting the enzyme, or it's not cutting the enzyme, it's cutting the substrate into two parts. And so kind of some features about aldolase is that it is a tetramer, alpha beta barrel enzyme, likely open sugar. So what happens is that going to step 4, we have this fructose 1,6-biphosphate. And from this, aldolase opens up this chain, or opens up the fructose 1,6-biphosphate, and that's how it's allowing the fructose 1,6-biphosphate to then break into two different parts, the dihydroxyacetone and the glyceraldehyde 3-phosphate. And you see does it have a high Gibbs free energy, so it's also be reversible. And so the rest of these bullet points kind of talk about the reaction mechanism as far as how aldolase does its thing with fructose 1,6-biphosphate. And these slides depict the arrow mechanism of what aldolase does. So I just want to make sure I got everything.
Oh, yes.
So it just contributes to cancer cell proliferation.
Yeah, that was a really good, really smart. It's high, right? Yeah. 141 that's 358.
So we also, I think that's it for that one. Does anyone have any questions about steps 1 through 4 before we move on to the second half? I'll kind of show you real quick. I use Anki for some of my cards when I try to memorize glycolysis. So I'm just going to show you real quick about several cards that I made up. I don't know, maybe it'll help. Some cards that kind of help me memorize what the enzymes were in order. I made this card like what are the enzymes used in glycolysis in order. I put each of the enzymes, and I kind of put like a brief function of them. This is kind of just overall just very general. I'd recommend kind of making cards of the specifics of the enzymes and the special features, because I know that Dr. Graves will test you on what the enzyme does, but these are just some basic cards that I made right here. We talked about upregulation, downregulation. So we'll go ahead and move on to 14.2. That will be the step 5. If anyone has any questions throughout the review, just let me know. You can put it on the chat, or you can also speak up as well. So triose phosphate isomerase. So we have another key here, isomerase. What I also like about enzymes usually is that in the name it kind of tells you almost like a clue as to what it does. So step 5, well, at least, sorry, let's go back to step 4. Step 4, from the fructose 1,6-biphosphate, we create 2 products, and that's dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now, glyceraldehyde 3-phosphate is the only product that can move onward or that can move forward in glycolysis. Dihydroxyacetone phosphate needs to be converted to glyceraldehyde 3-phosphate. And it does this through the enzyme triose phosphate isomerase. Now we see here, okay, we have like an isomerase here, and we have a triose phosphate. So we kind of know triose phosphate, that always kind of clicks to me, or kind of, helps me kind of know, okay, we're doing with like a 3rd phosphate, I guess. Triose phosphate, okay, so we're going to be adding a phosphate onto the trio, or the 3rd carbon. So in this case, we have glyceraldehyde 3-phosphate. So we're converting triose phosphate isomerase. We're kind of just altering this this a little bit into glyceraldehyde 3-phosphate. That's what an isomerase does. And this kind of talks about kind of like what the intermediates are, and as well as the features of the isomerase. So this is the function of it. DHAP slightly favored, but converted to GAP, because GAP is depleted by the next step in glycolysis. It's just meaning that GAP will be moving on. So triose phosphate isomerase can either be a dimer or a tetramer, and the active site is also an alpha-beta barrel. I believe step 4, I believe the aldolase was also an alpha-beta barrel as well. And this bullet point kind of talks about how the reaction goes. Glutamic acid, 165 initiates the reaction as a general base. So it abstracts the proton. I believe, I don't think it gives the reaction mechanism in this site. But this bullet point talks about what happens in the reaction mechanism. And we can see here that if glutamic acid is modified to aspartic acid, it slows the reaction down. It does this by placing the carboxylate group 1 amp farther away, because aspartic acid is shorter, has a shorter chain than glutamic acid. Glutamic acid, I think, has an extra carbon on its chain, and so when you modify it, or when you switch it out with aspartic acid, it kind of shrinks it, so the reaction is a little bit slower. So then we also have the enediol intermediate, symmetric, easily reversibles. This reaction is reversible. So, oops. The alpha-beta barrel, it's just like a secondary structure to the enzyme. You won't have to know like a whole ton of, you don't have to like explain anything like why does this enzyme have to have an alpha-beta barrel. But as far as features of alpha-beta barrel, let's see if I can put like a picture or something of kind of like what we're looking at here. I guess it's kind of like a protective covering. Let's see, I'll show you a picture of it real quick. It's kind of like what an alpha-beta barrel looks like. It has overall like a round circling the barrel are like beta sheets, and I guess like here, like on the top is kind of shows like alpha helices. I think it just acts as like a protective covering, but I don't think you really would have to know like a whole ton about the enzyme or about like that specific part. I think it's just important to know like what the active site consists of or what it is. So so yeah. This is good to know as far as the triose phosphate isomerase. This is another like, special feature, I guess. So this 10 amino acid flexible loop or flap at the active site overlays the substrate after the substrate. So the substrate goes into the alpha beta barrel, which is the active site, and then this flap kind of overlays the substrate. And it's able to, the purpose of this is to stabilize the ene-diol intermediate. I don't know if you can like visualize that. You have just like a barrel. I guess it's kind of like acts as like a covering or something over the substrate. And so it's kind of what triose phosphate isomerase is. All right, now we're on to step 6. Glyceraldehyde 3-phosphate dehydrogenase. Remember, dehydrogenase is an enzyme which removes a hydrogen. That can always be a key to determining what type of function it will serve. So in step 6, glyceraldehyde 3-phosphate, that's our substrate, dehydrogenase. We're removing this hydrogen, and we're substituting it with this phosphate group. And kind of in order to do that, we have an NAD plus molecule here that is getting reduced. Kind of how I mentioned earlier, this is one of the few, if not the only step in glycolysis that is a redox reaction. I don't know. I think it's the only oxidation reaction. I think it's the only redox reaction. I don't know if you guys know, correct me if I'm wrong, but I think it's the only redox reaction in glycolysis. It's the only one that involves NAD plus. And NAD plus is being reduced, so it's adding electrons, kind of taking away this. I believe NAD plus helps remove this hydrogen, and then this phosphate group then replaces it. And so this is also reversible. It has a large, I guess, smaller positive delta G. So, any questions about that? Okay. So obviously, yeah, we have the aldolase. This is kind of just an intro. Aldolase is, glyceraldehyde 3-phosphate. That's our substrate. So glyceraldehyde 3-phosphate. 3-phosphate is first oxidized by GAPDH. This is just glyceraldehyde 3-phosphate dehydrogenase, and an NADH is formed. Then it's phosphorylated by the same enzyme to form a high energy molecule that generates ATP in step 7. So it first gets oxidized by the NAD plus molecule, and so this NAD plus molecule removes this hydrogen, and then the enzyme allows the phosphate to then bind to the substrate. And it's a tetramer, and this allows NAD plus bound to be bound to 2 subunits. Oxidation phosphorylation. Remember arsenate, this was on one of the quizzes. I really think we should go much into that. And this is kind of the overall reaction, and this is the reaction mechanism for this. And here's NAD plus moved down to one of the
Hey, Josh,
the enzyme.
Technically, you're not supposed
should we
to
be
go
able to
back, but
draw
I'm
the
gonna
structure
throw
of
buddy,
NAD
if you
plus
will.
and NADH? Oh, somebody.
I'm saying I don't care.
Oops.
But,
Let's see who's.
yeah. Like a normal place.
All right. Sorry about that, Ren. What were you saying?
Yeah, sorry. I wanted to know if Dr. Graves will require us to know the structure of NAD+.
I don't think so. You won't have to draw that out or anything. As far as the test goes, as far as what I've seen, you don't have to know that.
Sounds good. Also, we're about to hit slide 12, and I didn't really understand what the purpose of slide 12 was.
Mm-hmm. Yeah. So this one, so slide 12, so yeah, thiols is at the active site and important for activity was evidenced by iodoacetate and activating enzyme. This was a, I believe this was like a study or something, and it's just determine the role of the cysteine, which usually creates, the thiol. And in this reaction, a histidine activates the thiol of cysteine. So the histidine removes or deprotonates the sulfur on the cysteine, and cysteine is a part of that active site in glyceraldehyde 3-phosphate dehydrogenase. And the sulfur then attacks the carbonyl to form the oxyanion, which then collapses back to reform carbonyl, and it transfers this this hydrogen, or sorry, the hydride to NAD+. So I'm going to see if this kind of shows it. Yeah, so right here, what activates the cysteine right here what activates the cysteine is this histidine deprotonates the sulfur, and then the sulfur then attacks the carbonyl, and then this is actually because of sulfur's electronegativity. Sulfur is a pretty electronegative atom, I believe. It's on like the top right-hand corner of the periodic table. But the sulfur then, once this the electrons then collapse back, then this hydride forms. A hydride is a hydrogen atom that has its shell filled, and so it becomes like a negative proton, or I guess a negative hydrogen, you'd say. So this is pretty highly electronegative. So then it attacks NAD+, and that's how NADH is formed. So that's kind of what the purpose of the sulfur is. But this slide is just to kind of determine what the role of it was, and they just use iodoacetate to determine what it was. So does that make sense?
Yeah, that was helpful. Thanks.
Okay. Step 7, phosphoglycerate kinase. Once again, this is a reversible reaction, but it's more kind of leading towards, the creation of ATP. We have 1,3-biphosphoglycerate, which is our product from step 6. And then it combines with ADP with phosphoglycerate kinase. Remember, we have kinase. So we're adding a phosphate group onto something. So in this case, we're adding it onto this ADP group here to form ATP. And let's see, enzyme mechanism analogous. So the mechanism, this is just another key here saying the mechanism is pretty similar to the hexokinase, but in the reverse mode. So that's another clue here saying, like, honestly, the amount of, I know like 10 enzyme mechanisms, it's kind of, it seems pretty daunting, but like around like 4 or 5 of them are really similar to each other. They're either like adding a phosphate group or subtracting them, and one of them's going one way, then the other has to go the opposite way. So, and then we have phosphoglycerate mutase. Now in this case, we didn't really talk. I guess mutase is just, it's just a regular enzyme. I mean, phosphoglycerate mutase, you're just transferring this phosphate group onto the second carbon of phosphoglycerate. So, and we have 3-phosphoglycerate, and that's our substrate for this, and that's our product from step 7. We're just converting it to 2-phosphoglycerate instead of 3-phosphoglycerate. We're simply just moving the phosphate group. Phosphoglycerate mutase is also a dimer, and the phosphate that's supplied is by BPG, and the mechanism kind of talks about, at least the mechanism for this enzyme kind of talks about what happens. It's just a really simple move over of the phosphate group. So in this first step, you just have a histidine that's bound to a phosphate group, and you have another histidine. This histidine deprotonates it, adds the phosphate group, and this deprotonates that histidine, and there's just like a switch over. That original phosphate group then goes back. So they just like switch places. And then we have our good friend 2,3-biphosphoglycerate, who we know very well from the first midterm. I won't go too much into detail on that. Then we have step 9, the enolase, and we take 2-phosphoglycerate. In this case, enolase is also a reversible reaction, and this is one. There's not really a whole lot of key words for this enolase. That's just one that you may just have to remember that enolase is in step 9, and it's just like a simple water, we're moving water to form an alkene here with this phosphate group on the second carbon. And enolase, has 3 dimeric subunits. So you have 2 alphas, 2 betas, and 2 gammas. And then this is just the active site. This is the magnesium ions. Magnesium ions just stabilize the phosphate groups, and because phosphate groups are pretty highly negative, and so it's just important to kind of recognize that. They also, in this case, they also stabilize this negative charge on the glycerate itself. Active site is also alpha beta barrel, and this just talks about the reaction mechanism. I would know this mechanism. Just review it and just determine to see like, okay, like this reaction mechanism involves a lysine and a glutamic acid. Fortunately, it's just 2 steps, so not super intense. And then our final one is step 10, which is the pyruvate kinase. And that one also involves the magnesium and the potassium, which stabilizes these negative charges. Pyruvate kinase, it's also irreversible. As we see here, it has a large negative Gibbs free energy. And it's also analogous or very similar to the hexokinase in reverse. So this step 10 is also similar to how step 7 is. And so that's also comforting to hear as far as the mechanism goes.
Josh, just a question.
Yeah.
So basically steps 1, 3, 7, and 10 are basically the exact same thing, just some are going forward and some are going backward.
Yeah, exactly. Steps 1, 3, 7, 10. I think enolase is unique. Step 8, step 8's also unique. And step 7 and step 6 I know is mostly it's just step 1, 3, 7, and 10 are the most similar. But they each, steps 1 and 3 go one way, and then steps 7 through 10 go the opposite direction. So pyruvate kinase, kinase, we add the phosphate group onto ADP to form ATP, and these are also, here are some more factors that upregulate, and these are some that downregulate, and these are different, these are the different isoforms, which Step L, liver, M in muscle and brain. So that's kind of key there. M for muscle, L for liver. We have here, low glucose leads to increased CAMP. Cyclic, I think that stands for cyclic adenosine monophosphate, which then leads to phosphorylation and activation of the enzyme. So this is kind of a regulatory mechanism that we have in our bodies. So if our bodies don't have a lot of glucose, then obviously glycolysis wouldn't be going on, and this is kind of explains why glycolysis, or kind of a regulatory step as to why glycolysis doesn't happen. The low glucose leads to increased cyclic AMP, which then leads to phosphorylation of pyruvate kinase, which inactivates it, so that glycolysis doesn't continue. And here's just it's just pyruvate in the different forms. And pyruvate's a key transfer molecule. Now in chapter 14, chapter 14 says, know the anaerobic lactate dehydrogenase reaction. So this slide pertains to that step. And I know there will be a question on that one. Under anaerobic conditions, NAD plus is required for step 6 of glycolysis. So under anaerobic conditions, the only process that really occurs is glycolysis and this reaction here. Because under anaerobic conditions, we need energy, like really fast. Usually aerobic conditions, we have oxygen where the pyruvate's able to continue going on to different processes like the Krebs cycle and the electron transport chain, and those generate abundant amount of ATP. But under anaerobic conditions where we don't really have that resource, we don't have oxygen, so pyruvate, glycolysis needs to go, but in order for glycolysis to go, it needs NAD plus, and that's the ultimate purpose of this reaction, is that we need to generate NAD plus so that glycolysis can continue to move onward. And the way that it does this is through converting pyruvate to lactate, or lactic acid. And the lactate dehydrogenase is the enzyme which which allows this whole reaction to happen.
Do we need to know any mechanisms behind that, or just that it actually happens?
No, just that it actually happened and the purpose of it.
Cool.
And so the ultimate purpose is to regenerate the NAD plus, so that glycolysis can continue. Just moving on to the next slide says no feeder pathways, steps, intermediates in the individual fructose paths, intermediates of glycolysis formed from nanocircle lactose. So this is a good diagram, kind of describing the different feeder pathways from the different sugars. Since ultimately our diet consists of sucrose, lactose, all these different sugars, and this describes how this reaction kind of plays into glycolysis. So in the case of fructose, fructose is perhaps the most complicated one to know, at least as far as how fructose enters the glycolysis. Fructose here, fructose converts to fructose 1-phosphate, and it's able to do that. Sorry, I kind of lost track here. We have two different ways where fructose can enter glycolysis. We have one that involves hexokinase, and this reaction occurs in the muscles. Muscles and the kidney just involve simple hexokinase, and it adds a phosphate group onto fructose, and it creates fructose 6-phosphate, which is immediately goes into, it's a part of glycolysis. When we add fructose to glycolysis in the liver, however, it's a little bit more complicated, because we have to involve various different enzymes. Hexokinase isn't really available in the liver, at least like with the hexokinase that's being used for fructose. So fructokinase is that enzyme along with ATP, to add a phosphate group onto fructose, but instead of the 6 carbon, it adds it onto the first carbon. So it converts to fructose 1-phosphate. And the aldolase of that, we can, use that, I'll use the keyword aldolase, we know it cuts it in half into glyceraldehyde or dihydroxyacetone phosphate, and then dihydroxyacetone phosphate. We know that from glycolysis, that obviously the triose phosphate isomerase just converts it to glyceraldehyde 3-phosphate. And then glyceraldehyde, fortunately, we just need to add a phosphate onto there to form glyceraldehyde 3-phosphate. Yes, Nate.
Sorry, I was trying to have that feature. Do we need to keep track of the energy used in these feeder pathways?
Like, kind of like
ATP wise, like net in, net out, all that stuff.
I don't think so. As far as like the feeder pathways, you won't be tested on a question saying like, oh, what was like the net ATP if you were to use mannose, or if you were to use galactose instead of something else. I'm gonna double check that real quick, but I don't think he would ask you for that. Is that what you're referring to?
Yeah, just some sort of question along those lines, whether it would ask for if ATP is used in some sort of step and how much.
Mm-hmm. I know you have to know that for glycolysis.
Yes, yeah.
But let me double check that real quick.
So Josh,
Mm-hmm.
I'm not gonna lie, this is one of the slides from this unit that has confused me the very most.
Right.
Um,
That's
just
totally fine.
it's really dense, I feel like. There's just a ton of stuff in it.
Yeah.
So like what's an example, do you think, of something he could ask about on this? Like what are the main points that we should kind of gather from all this?
I could see him like kind of just, I guess, like asking like what enzyme would involve fructose. He won't give you a diagram and ask you to like fill in the blank of it, or fill in the blank. Let's see here. I'm just double checking the kind of the notes that I have here.
Can I mention something he said in his office hours today?
Yeah, of course.
So I just went to his office hours today, and he said the fructose pathway, you need to know the enzyme and the intermediates, but only for fructose. The other ones, like glucose, which is obvious where that comes in, but galactose or mannose, you just need to know where it goes into glycolysis. So like for D-galactose, you don't have to know that it turns into UDP glucose, UDP galactose being UDP glucose, and then glucose 1-phosphate. Like you only need to know that stuff for the fructose pathways.
Mm-hmm. Yeah.
So what did he say that we needed to know for D-galactose, for example? Like where would it go in?
Just know that it goes in as glucose 6-phosphate. Like it enters glycolysis as glucose 6-phosphate. And then for mannose, you would need to know it enters as fructose 6-phosphate.
Gotcha. Okay, that's very enlightening. Thank you.
Yeah.
And so we should also know which step that is, right? That like the galactose enters at step 2 as glucose 6-phosphate. Is that right?
You won't have to mention the step. Well, I mean, you will like know what the steps are just for like the actual test. I mean that's pretty. I mean that's pretty simple. Just like if you see, okay, glucose 6-phosphate. Oh, that's in step 2. Usually, when I had the midterm on glycolysis, the first thing that I did when I entered the testing center was that I quickly wrote down, or I quickly did like a basic outline of glycolysis, what the enzymes were and what the substrate and products were, so that I can have at least that as a background. And I believe that I also wrote down some of the more complicated mechanisms, like step 4 with aldolase, Step 6, I believe those were 2 reaction mechanisms I wrote down, because those were some like the more complicated reaction mechanisms. But simply like immediately, like before you start the test, writing down what glycolysis is and what the enzyme is. That way you don't get like messed up when the test, when you're involved in the test. So,
Can I
and
ask a clarifying question?
yeah,
So on this slide, basically like how to read it is if you're I may be totally off, so just tell me. But if you're looking at like lactose, you're saying that lactase turns it into degalactose,
mhm.
and then you would go from degalactose, and you would say that though, I mean, there's no enzymes there technically, but like, what kind of question would he ask where you would need to know like degalactose goes in, would it be like goes into glycolysis at glucose 1 phosphate? Is that kind of what it would be asking, or like?
Yeah, kind of. Who's the one that that talked with Dr. Graves? Was that Isaac? Who talked with Dr. Graves today about that? Was that
About what?
one of those
I
guys?
talked to him today about this.
Yeah.
What was?
What did you tell you? Kind of. She just asked like what's like important to know, like with galactose and
Yeah.
with the pathways with that.
Is it possible for me to annotate? Because that might help.
Yeah.
Is that a possibility?
Let's see. Hopefully.
I think I can. I just want to make sure.
Okay. Try it. I don't know.
Okay. Okay. So for this, you need to know everything that is contained in this like circle. Well, for fructose, okay? And then for like galactose, you don't need to know this. You don't need to know this. And I don't think you really even need to know this. But you do need to know that it comes in as this, as glucose 6-phosphate. And then for mannose, you don't need to know this. You don't need to know this or this. You just need to know that it comes in as fructose 6-phosphate.
Okay.
And
So,
if something like that shows up on the test, that it was totally Dr. Gray's fault because he told me that is not going to be on there. So, but this part, like fructokinase turns it into fructose 1-phosphate, and then fructose 1-phosphate aldolase turns it into glyceraldehyde. You need to know all of those specifically, and specifically that happens in the liver. Like these bottom ones happen in the liver, and hexokinase right here just happens not in the liver, just anywhere, I think. But the bottom part, this whole complicated part, you need to know. So specifics about it.
okay, cool, thanks.
Yeah, no problem.
Yeah, great. Yeah, that was great explanation. Yeah, just the fructokinase, that whole reaction is the liver. Hexokinase is muscle. I drew this one out because this one seemed a little bit more complicated, but again, looking for clues of the enzymes that are involved. Fructokinase, we look at phosphofructokinase in glycolysis, and that one, in that reaction, we add a phosphate group to the first carbon. So we have fructokinase adding fructose to the first carbon. So we have fructose 1-phosphate. And then just remember, we just have an aldolase that splits it into 2. Glyceraldehyde is one product in dihydroxyacetone phosphate. That's a product, or that's a substrate. That's one of the products that we get from step 4 in glycolysis, and we just convert that to glyceraldehyde 3-phosphate. And glyceraldehyde, we just simply need to add a phosphate. And the enzyme triose kinase adding a phosphate group, triose, third carbon, trio, just looking at different keys like that. So, and then, does anybody have any questions about the feeder pathways? I know that one's a little bit.
Yeah, if we jump forward a few slides when it shows the entry of galactose into glycolysis, so like we don't really need to know any of that then, right? Like the next slide after that,
This
or
this one.
Oh, that one. Yes, that one right there.
This one.
We just need to know that enters as glucose 6-phosphate. We don't need to know all of
Yeah,
it.
no, we don't. No, you don't need
Okay.
to know this.
Perfect. Thank you.
Yeah, I'd feel very bad. I feel very sorry. Yeah, no, you don't have to know that. Yeah, just exactly what like Preston said is good. All righty. Okay, so we know that the feeder pathways. I'm going to take a quick bathroom break. It only take about like 3 minutes or so, but if you have any questions about the gluconeogenesis and the oxidative pentose phosphate pathway, just let me know. Just write them down, and we'll quickly like review those, and then we'll get into chapter 15. I know chapter 15 involves a majority of other questions, so we'll quickly get into that. I'll quickly share chapter 14 real quick. 14.3 here. And I'll talk about gluconeogenesis. Okay. All right, I'll be right back. Okay. Here we go with gluconeogenesis. Okay. Does anyone have any questions before I dive into gluconeogenesis and into the pentose phosphate pathway? Does anyone have any questions about specific parts of gluconeogenesis? Okay. We'll just quickly go according to the pointer here. Okay. So for gluconeogenesis, we just need to know the substrates, the products, enzymes, and locations of the bypass steps 1 and 2, which I'll quickly review, or we'll review and talk over about that, and know how reactions take place generally. But you do not need to know any mechanisms for this. Also kind of referring to special requirements, cofactors, and requirements for glucose transport. So gluconeogenesis occurs mostly in the liver. That's where like a majority of it happens. But it is also like there's also like slight possibilities of gluconeogenesis occurring in like the kidney, brain, heart, muscle, things like that. And it usually uses pyruvate lactate, oxaloacetate, or amino acid remnants in order for gluconeogenesis to occur. Fortunately, gluconeogenesis is just kind of a backwards process compared to glycolysis. The differences are the initiation, the first two steps of gluconeogenesis, and I believe opposite reactions to the two irreversible reactions, step 1 and 3 of glycolysis. And those are just the main 4 enzymes to know. Step 9 and 11, all you have to know is just what the enzyme and the and just like the substrate products of them are. And that's it. You don't have to know anything more about that. The key with gluconeogenesis is mostly understanding where the 2 bypass steps are, and what happens with that. So with gluconeogenesis, since gluconeogenesis is the opposite of glycolysis, and we're mainly going to talk about how pyruvate is converted back to glucose. Pyruvate is moved from the cytosol into the mitochondrial matrix. So at the end of glycolysis, we have all these pyruvate molecules that's in the cytosol. But in order for pyruvate to convert back to glucose, it needs to go into the mitochondrial matrix. And in the mitochondrial matrix is this enzyme called pyruvate carboxylase. And we see here that in both of these conditions, pyruvate carboxylase is that key enzyme which converts pyruvate to oxaloacetate. And oxaloacetate is a molecule that's later involved in the citric acid cycle and things like that, but you don't have to know that for this test. I'm just kind of giving you some context behind what oxaloacetate is. I'm going to talk about real briefly the differences between those two pathways. And yeah, so it's converted to oxaloacetate. Oxaloacetate most of the time is converted to malate by malate dehydrogenase, and that's another enzyme that's in the mitochondria. And it's able to be exported to the cytosol by the malate shuttle. This describes what's going on with the malate side of converting back to glucose. However, if lactate is high, then most tissues generate oxaloacetate, and then converted directly to PEP, or the phosphoenol pyruvate, in the mitochondria by the carboxykinase, and then export to cytosol. So that's a whole lot of words. I like visuals. Visuals just kind of just help me a lot better understand what's going on. So we'll first talk about malate here. So we have pyruvate. Pyruvate is in the cytosol, but under conditions that facilitate gluconeogenesis, we need to have pyruvate be converted to oxaloacetate. And in both conditions, oxaloacetate is formed in the mitochondria. But the thing is that oxaloacetate needs to go out into the cytosol. And oxaloacetate, there are no transporters in the mitochondria for oxaloacetate to exit the mitochondria. So in the malate condition, or the malate situation, it needs to be converted to malate, and it's able to do this through the malate dehydrogenase. And in this case, as a part of that condition, we generate NAD plus. And once malate leaves the mitochondria, then it's able to be converted back to oxaloacetate, and oxaloacetate then moves on to the remaining steps to then be converted to PEP. Now, when lactate's high, so when we're having, when we're under, at least like we have like a lot of lactic acid, and we want to get rid of all that lactic acid, our body tries to get rid of it. And a way that it does this is by converting lactate back to pyruvate. And we remember from our lactate dehydrogenase discussion under the anaerobic conditions of the reasoning why the lactate is there is to regenerate NAD plus molecules. So in this case, we're doing the opposite. We're using NAD plus molecules to then create pyruvate. And this generates NADH molecules, or NADH, yeah, NADH. And then pyruvate is then able to enter the mitochondria. It's converted into oxaloacetate, and then converted to PEP. And there are transporters in the mitochondria for PEP. And PEP just then moves on to the rest of the steps in gluconeogenesis. Does anyone have any questions about that? That covers a majority of what's going on with gluconeogenesis.
Can I ask a question about what it means when it says bypass steps 1 and 2? Can you just remind me what that means?
Yeah. Yeah. So when it's referring to bypassing, so it says especially enzymes and locations of the bypass steps 1 and 2. So this refers to the bypass steps because we're bypassing, at least we're kind of using an alternate, at least 2 different like alternate methods to create phosphoenol pyruvate, and those are like the main two steps. Let's see, steps 1 and 3 of glycolysis are bypassed with single alternative steps. So what's the bypass steps just kind of like, it's just like a way around the steps, around the later steps of glycolysis, if that makes sense.
Yeah, that's helpful. Thank you.
Okay.
Yeah. Is there a slide in this PowerPoint that shows all of gluconeogenesis in like just in the big picture of it all in one slide?
Yeah. Let's see here. I don't think so, honestly. Here, I'll bring up a picture real quick.
I'm just having a hard time seeing where the steps are broken up in each slide.
Right.
You know?
Yeah. I don't blame you.
I don't believe there is one. You might just have to like look it up online or something.
Yeah, I don't see any on this slide.
Because it doesn't even really say in the slide which steps they are, right? Like which steps are happening.
Yeah, here I'll label which ones are irreversible or not, because there's four main steps here. Let's see if I have another. I made a card on this once. Let's see if I still have it.
What about like slide number 5? Doesn't that show like the gluconeogenesis pathway, how it comes back around? It just doesn't go into depth of everything else within those 2 steps.
Yeah, it shows the first couple of steps. Yeah, the first part of gluconeogenesis. So the first 2 steps that we're talking about were these 2, the pyruvate carboxylase and the PEP carboxykinase. And then these steps are all the same in glycolysis. So we still have phosphoglycerate. That's from step 8. But we see a PEP, and then this is reverting back, phosphoglycerate, phosphate. Pretty much all of what kind of helped me was that all of the enzymes and steps that were reversible were the same in gluconeogenesis and glycolysis. The steps that were irreversible in glycolysis needed to have bypass steps in gluconeogenesis because you're trying to bypass the irreversible step. So steps 1 and 3 in glycolysis both needed to have bypass steps, and so did step 10. And step 10, in order to create the phosphoenolpyruvate, in order to bypass step 10, which is the pyruvate kinase, we need 2 steps to go back to phosphoenolpyruvate. And then here are the 2 bypass steps from steps 1 and 3 of glycolysis. Each of those results in at least in gluconeogenesis case, both result in phosphate losses. And these are the two specific enzymes. Fortunately, it's not too difficult to kind of look at the enzyme and think, oh, this is completely different. Both of these enzyme names both involve the substrate, so it's really easy to remember saying, oh, well, you're just using fructose 1,6-biphosphatase. That's your enzyme to convert this to that, and then glucose 6-phosphatase. So fortunately, the biochemists were pretty merciful on us in this case with naming them. You won't have to know this. Let's see. You will. I think the only thing you have to know about this is that biotin is involved. See the special requirements cofactors with pyruvate carboxylase uses biotin.
Hey Josh, I don't know if it's just me, but I think you have to like switch your screen over. I think it's just showing.
Oh, sorry. My bad. I'm so sorry. I was just referring to this slide right here with the first 2, or the first 3, the first step of glycolysis and the third step, and these are just 2 bypass ways. That's just what I was referring to. And as far as this, you won't have to know this reaction mechanism. You just need to know the parts of it, or at least the cofactors, I guess.
Do we need to know like what it's named each time it gets attacked by something? Because I don't really know what the names are. I just know like what happens, but I know like the name of the beginning, like oxaloacetate. But do we need to know like the intermediate names? Does that make sense?
Like in this case with the reaction mechanism?
Yeah, like the name of the intermediate.
Or these?
No, the one you had before.
The one I had before. No, you won't have to know. You just need to know like what the cofactors are in this case. So we have in converting, let's see, pyruvate carboxylase uses biotin to bind the phosphobicarbonate. And this is just the formation of oxaloacetate from pyruvate in the mitochondria. And so it just uses, in order for oxaloacetate to form, it just uses biotin and the phospho and this bicarbonate reaction here. It uses ATP in order for this to happen. So I believe that's all you need to know for that case. Okay. So yeah, you just need to know the substrates of that pyruvate carboxylase. So we have bicarbonate, we have ATP, we have bicarbonate, we have ATP, and then we have biotin. Oh, sorry, pyruvate. bicarbonate, ATP, pyruvate, those three. Those three substrates. Biotin is just kind of an assistance to it. Okay, so what we have here is then just the phosphate pathway. Real quick, slideshow 14.4. That's your slideshow. I'm not gonna go really because it's only one question. I'm just gonna trust you all to just know the products of the pentose phosphate pathway. Just what's created. Overall, it produces ribose, NADPH, other pentoses, but that's very, very broad. I would just recommend you all just looking at slideshow 14.4 to review the oxidative side of the pentose phosphate pathway, because there's an oxidative and an oxidative, but just review mainly the oxidative pentose phosphate pathway. Chapter 15. We're going to have to cruise by on this one real quick. There we go. Okay, I'm just trying to find 15.1 real quick. Here we go. Okay, so we have 15.1 here, and with these questions, it's just, or at least with this section, 15.1 talks a lot about the regulation of glycolysis and gluconeogenesis. And the pointers on the study guide says including glucokinase, fructose 2,6-biphosphate, xylulose 5-phosphate, and PPA2. I know the effects of hormones. So with that case, with glucose metabolism, it's important to know what insulin does, what enzymes insulin affects. That's at least as far like the hormone signs go. These next two slides are also important to review. The hexokinase 1, what differs between hexokinase 1, 2, and 3. What are they inhibited by? And hexokinase 4, which is in the liver, glucokinase. It's highly sensitive to glucose concentrations. And then, yeah, just hexokinase 1, 2, and 3. I know I think there will be a question or something on that. PFK is highly regulated. Let's see.
Are we required to know the difference between 1, 2, and 3, or just what they do?
Just what they do. Most of those, I think those can be, you're safe to assume that those three can be combined into like a group because all of those three are really similar. Mostly it's just the differences between
Non-hepatic.
Yeah.
Okay.
Yeah.
Cool.
This fructose 2,6-biphosphate, this is important to understand that it's hormonally regulated. So insulin plays a role in this with this factor as well, with the hormones. This is one of the key slides that's important to know with fructose 2,6-biphosphate. It's a primary reciprocal regulator of glycolysis. I believe it activates glycolysis. So this slide's really, really good to know with as far as the hormonal regulation of fructose 2,6-biphosphate. So under high glucose leads to increased insulin, and this leads to the dephosphorylation of that complex, which leads to the increased phosphofructokinase 2 activity, which then increases fructose 2,6-biphosphate, which increases glycolysis. And this can also play a role in inhibiting, well, as a part of activating glycolysis, it inhibits gluconeogenesis. As far as those enzymes, let me double check to see how much you. Yeah, it's important to know what happens with this case, what activates this complex. So we know that dephosphorylation by PP1 of this complex leads to increased phosphofructose-2 kinase activity. So phosphorylation does the opposite, so it shuts it down, and that reduces the fructose 2,6 biphosphate. And as a result, it does the opposite of what this step does, which is stimulating gluconeogenesis. So this slide's important to know. This one includes a lot of bolded points. Xylulose 5-phosphate, which is a side product of the pentose phosphate pathway, and this activates PP2A, and PP2A dephosphorylates target proteins. Just like this base, which we were referring to here, and this activates F26 biphosphate. So just to review, xylulose 5-phosphate activates PP2A, and PP2A dephosphorylates this complex, and that complex allows more production of fructose 2,6 biphosphate. So indirectly, xylulose 5-phosphate stimulates glycolysis. That's what we can learn from this. By this process. So it shifts balance towards glycolysis, acetyl-CoA production. Also, high phosphoenol pyruvate inhibits PFK1. So this is a factor that downregulates glycolysis. This phosphoenol pyruvate is one of it's our product from step 9 in glycolysis. So from this, because we have a high product value, that will cause down regulations or a negative feedback loop to decrease glycolysis. So that's kind of key with knowing a lot of the factors that upregulate and downregulate glycolysis and gluconeogenesis. If you have a lot of product of something, then it will cause downregulation. If you have a lot of reactant, then it will cause overall an upregulation of like a process. So let me see if I covered everything on that. So we had glucokinase, and that was hexokinase 6, fructose 2,6 biphosphate, that effects of hormones. Okay. I think we covered everything there. We'll go to slideshow 15.2 real quick. And I think that one talks a lot about the glycogen. Oops. Okay, so glycogen. We all know that glycogen, it's stored in the liver. And greater concentrations in the liver, 10% of wet weight. But there is also like a good amount in muscle as well. And it allows glycogen, it's usually some of the most immediate source of instant glucose that we can get. So when we're fasting, when we're fasting, usually our body reverts to glycogen in our liver as immediate sources of energy to last us a majority of the 24 hours if we're fasting for 24 hours. And it's important to know that it's mainly alpha-1,4 linkages, but there is branched over every 8 to 12 sugars from alpha-1 to 6. That was a part of one of the things we need to know. So that's just a building block of glycogen. As far as glycogen goes, we just need to know the enzymes, locations, Steps involve substrates, products, glycogen, glycogen synthase roles, branching, how glucose is exported. So this involves 10 questions. So I would pay attention to this as part of your study, since it does involve a good chunk of the test. So this slideshow 15.2 will be very helpful with that. So we'll just go ahead and quickly kind of cover what glycogen synthesis is, and then hopefully we'll talk about this for the next hopefully like 7-10 minutes, and then we'll finish on the last pointer of these regulations. So that's the building block. Sugar nucleotide UDP contributes to glucose 2 glycogen synthesis. So glucose 6-phosphate is converted to glucose 1-phosphate by the phosphoglucomutase, and that's combined with UTP to form the UDP glucose. A phosphate group leaves, and then leaving behind pyrophosphate equivalent to hydrolysis of 2 ATP to 2 UDP. I'm just trying to figure out to see what the purpose of this one is. So we just have a fructose, a phosphate group that this is NTP. This produces all the sugar. That sugar nucleotide contributes glucose to glycogen. So this is glycogen synthesis. UDP glucose is the substrate for glycogen formation. So in this case, we're talking about forming glycogen. So in formation of glycogen, the first glucose needs to be coupled with glycogenin, and that's the key there. So glycogenin, you have a source, that's glycogenin, and glucose molecules are coupled, or glucose molecules bind to that, and that's kind of allows it to stay there and has an anchor point, and glucose molecules are then added onto that. 6 to 7 additional glucoses are added to glycogen chain. So sometimes glycogenin can also add, or I guess glycogenin adds as glucose molecules themselves. And then later glucoses are then added by glycogen synthase, which is an enzyme which, builds up the storage. And we already know this from our earlier slide. And this is a depiction of glycogen, and you won't have to know like secondary structures or anything like that. So we know that acts as the anchor point in glycogen, and then glycogen synthase adds more glucose molecules onto there. You won't have to know the reaction mechanisms for this, but it's important to know what the steps are and the enzymes involved with it. So in this case, addition of first few glucoses to a tyrosine. So what we have here, we have a regular glucose molecule here's our glycogenin, which has a tyrosine on the end. Glycogenin is the protein and it's tyrosine. Then you have your glucose here, which is bound to a UDP molecule. And UDP is kind of our leaving group in a way. Kind of allows this tyrosine to be able to attack glucose here. And then UDP then leaves, and okay, sorry, I just got confused for a sec. Okay, so glucosyl transferase, so glucotransferase, we have a transferase here, which, as we know, transferase is our key word in this enzyme to understand what it does. A transferase, moves functional groups off, and so that's what allows UDP to leave and allows glucose to officially bind to this glycogenin. So the glucosyl transferase is a good, it's an important enzyme to know. It's just an initiatory enzyme which allows the first glucose molecule to bind to glycogenin, and then glycogen synthase then takes over its role. And then glycogen synthase is that's our continuing enzyme, and it allows the alpha-1,4 linkages, except every 8 to 12 sugars where you have the 1,6 branch point, which is depicted right here. And so now we're going to talk about real quick glycogenolysis, which is lysis is always, that keyword lysis depicts everything that's breaking down. So glycogenolysis, we're breaking down glycogen. Glycogen is cleaved by phosphorylasis. So in this case, the phosphate is able to activate. I'm better with kind of describing what's going on by the picture. Is there a picture here? Yes, there isn't. Okay, well, let's go off here. Glycogen phosphorylase. So we have glycogen. We have this phosphate groups here. Let's see, the phosphate on the peroxidyl phosphate activates a phosphate in the solution, which attacks and then cleaves the glycogen alpha-1,4 bond to generate glucose 1 phosphate. And so we have right here. And it only works at non-reducing ends of glycogen. So at the very end of a long chain of glucose, we just have the 4th carbon, and then it just removes glucose molecules. More ends that we have, there's faster release. Reaction is reversible, so delta G is pretty close to 0. So we have, as glucose is being broken up, we have glucose being added onto glycogen. And then glucose 1 phosphate is then converted to glucose 6 phosphate. And then, of course, we know that glucose 6 phosphate goes into step 2 of glycolysis. And here's peroxidyl phosphate, which activates the phosphate group. You won't have to know, or you won't have to draw out the peroxidyl phosphate. And then the glycogen phosphorylase, we have here phosphorylase, which is we're removing a phosphorylase. So a phosphatase removes a phosphate group, but a phosphorylase is an enzyme which allows a phosphate group to attack and to break off or cleave glucose molecules. So this is what allows the formation of glucose 1 phosphate. However, on glycogen breakdown, we have our phosphorylase. It moves down the chain pretty easily on the alpha-1,4, but when it encounters a branch, though, it's not able to do that. It's not able to cleave branches. So what it does is that it stops 4 residues away from the branch point, and then there's a debranching enzyme, which moves a group of 3 residues from the branch chain to the main chain. I believe this picture kind of shows what's going on here. So we have our breakage point right here. We have glycogen phosphorylase that's reducing, or not reducing, that's breaking down these glucose molecules, and then gets to the 4th glucose molecule from the branch, and then it allows this transfer of 3 glucose molecules, this debranching enzyme, and it binds it, or it allows it to then kind of shift to this group of glucose molecules, and then the debranching enzyme then just cleaves out this group right here, and then glycogen, and then the glycogen phosphorylase then continues its job with breaking down the rest of them. Does that make sense? Anyone have any questions about that? Okay. And so then our final step, or one of our final steps with glycogen to glucose, is phosphoglucomutase. This is an enzyme that's important to know because it allows glucose 1 phosphate to then be converted to glucose 6 phosphate, which then of course goes into glycolysis. And overall, the equilibrium lies towards glucose 6 phosphate. And this kind of describes what the mechanism is of that whole process is. I'm not going to go super deep into it. I believe these steps kind of describe what's going on pretty well. This is the reaction mechanism overall for phosphoglucose fructase. And then this is just kind of explaining that the situation skeletal muscles are kind of significant. They need special enzymes in order for this to happen.
Hey, Josh.
Yeah.
So on that last slide with the reaction catalyzed by phosphoglucomutase, where it yes, right there. So is this showing what we're doing to those glycogen products that we've knocked off the glucose 1-phosphate and just showing how we're converting it to glucose 6-phosphate so we can then use that in our cycle later on?
Yeah, exactly.
Okay.
Yeah, the reaction is just showing what happens with the transferring the phosphate to the 6-phosphate. Because we have the phosphoglucomutase, which has a serine, and that's bound to a phosphate group. And what happens is that the serine's phosphate group then binds to the 6 carbon on glucose, and then it takes away this phosphate group. So it's just kind of like switch it, I guess.
Cool. Thank you.
Yeah. So our final 10 minutes, we'll just talk real briefly about 15.3. And that talks a lot about different hormones, regulatory things, know about regulation of glycogen synthesis and breakdown, enzymes that phosphorylate, that whole last section. So what we have here is glycogen phosphorylase. The phosphorylase keyword here, phosphorylase, involves a phosphate attacking the glucose molecules, or attacking a glucose molecule so then it can then be cleaved or taken off of glycogen. And this enzyme is activated by phosphorylation and then inactivated by dephosphorylation. Let me double check this real quick here, see if this involves anything else. Yeah, so this kind of talks about the importance of pKa, glycogen phosphorylase, which is the enzyme that we were referring to in this slide. It's phosphorylated, which activates it. And then pKa's role is to phosphorylate phosphorylase kinase. So I would recommend drawing this out to kind of identify, okay, what activates what. So this whole thing, pKa activates phosphorylase kinase. And it does that by having it phosphorylated, and then phosphorylase kinase. Now kinase here, this is our keyword here, kinase. So it phosphorylates something, it adds a phosphate group onto glycogen phosphorylase, and that activates glycogen phosphorylase. So in this whole situation, adding a phosphate group onto something will activate it. That's pretty much the key with this whole situation.
So can you just say one more time, like, what is it that actually inactivates glycogen phosphorylase?
Yeah, so dephosphorylation. So removing a phosphate group from glycogen phosphorylase inactivates it. So so we just have pKa. Maybe I should do.
So I guess, like, what is it that dephosphorylates it?
Oh, I see.
Like, the name of that.
The name of that?
Yeah.
I am not.
I think it's like the next slide.
I'm not sure.
With the phosphorylase phosphatase. That one? Which is from the insulin?
Yes. See, insulin leads to BP1 activation, which causes dephosphorylation of phosphorylase, and that leads to their inactivation. So kind of like how I said before, know what insulin does, know what its role, because insulin has a pretty large range of what its roles are. You just kind of need to know like what aspects or how insulin affects these different processes. So here's an example of what insulin does. Insulin leads to PP1 activation, and that causes the inactivation of the enzymes. Those two. So, and then glucose. Obviously, that's a regulator of glycogen phosphorylase because it's a product of glycogen phosphorylase. So just kind of like if there's like a lot of glucose, then there's a lot of insulin, and that leads to deactivation of phosphorylase kinase and glycogen phosphorylase. So it all like interconnects with each other. There's always a reasoning why these things happen, and usually just leads to the conditions, the environment of the body, I guess. And then these couple slides talk about how glycogen synthase is regulated. In glycogen synthase, phosphorylation is inactive. So it's kind of the opposite of what we were going with as far as glycogen breakdown. A lot of glycogen breakdown, if you phosphorylate something, it activates it. But in glycogen synthase, phosphorylation kind of deactivates it. Or the phosphorylated form of glycogen synthase. And then here's just kind of repeating what we're saying. Insulin through PP1 dephosphorylates, and in this case, it activates glycogen synthase. Rather, in this case, insulin leads to PP1 activation, which causes dephosphorylation of all that, so that deactivates it, but it activates glycogen synthase. And then, I don't know how much, let's see, let me double check with GS3. This is another enzyme that insulin regulates, GSK3. So it indirectly regulates that. GSK3, let's see, do we need to know about that one? So we just need to know the enzymes that phosphorylate and dephosphorylate that, glycogen phosphorylase, phosphorylase kinase, glycogen synthase. Know the effects of hormones, know about the enzyme complex present on glycogen granules. GM effects of PKB and PKA. GSK3. I'm going to get back to you on that. I don't know how much information we need to know on GSK3. But I do know this slide's important. We'll just talk about this for the next couple of minutes. Regulation of PP1 by GM. So when we go back to PP1, PP1 is an enzyme that is activated by insulin, and PP1 just causes dephosphorylation. When PP1 is activated, it causes dephosphorylation of like everything. So in this case, it causes dephosphorylation of phosphorylase kinase, of glycogen phosphorylase, and also glycogen synthase. And so when we go back here, GM is phosphorylated in response to insulin. Insulin is abbreviated to PKB. And so this allows, so insulin, this allows it to interact with and activate PP1, which causes it to bind, PP1, like we're saying, it dephosphorylates things. And like how we talked about before, it activates the synthase by dephosphorylation. We talked about this. Epinephrine, this pointer right here kind of satisfies the point of saying, know the effects of hormones. Epinephrine is a hormone, and it leads to an increase in PKA. And as a result, it leads to phosphorylation of the GM. And then that results in the GM's dissociation from PP1. So GM, is bound to PP1. And then, and because epinephrine increases pKa, then and GM's disassociated from PP1, then that shuts down all PP1. And so as a result, everything's still phosphorylated. Everything's still going on. So, and this is just an overall diagram of what's going on with this whole this process. Ultimately, for 15.3 or slideshow 15.3, we just need to know how glycogen synthesis and breakdown is regulated, and just kind of those enzymes that we talked about, such as PP1, GM, how pKa and pKb affect those. I don't think you have to know GSK3. I'll send in an email if I do find out that you need to know about that, but I don't think you really need to know anything about GSK3. Mostly it's like these beginning slides that you just need to focus on. Just noticing like what phosphorylates what and how phosphorylation inactivates or deactivates something. So does anyone have any other questions? I know I just kind of just went on on a tangent. I just wanted, or at least I just wanted to make sure that I covered everything for the test. So does anyone have any final questions in regards to this or anything as far as what you should know for the test? Okay. Okay, awesome. I'll be holding office hours from 2 to 4 tomorrow. So if you find out any questions that you have about this information, you're feel free to come to my office hours tomorrow, and I'll be able to help you out. But I hope that the review was kind of helpful, and I hope you guys just continue just to study, make flashcards, just practice drawing things out, and you got it. So, and I'll be sure to upload this, or at least be sure to send you all the link to the slide, so, or to the review.
Thanks, Josh.
No problem.